Good afternoon. Welcome to Labcorp and Median Joint Webinar. We're happy today to bring you the topic on overcoming the challenges in deploying new imaging biomarkers in clinical trials. Perspectives from patients, physicians, and other stakeholders. New imaging biomarker has a strong potential to provide additional insights for trials, to better select patients, to predict response to therapy, to early assessment for efficacy and safety. But implementing those imaging biomarkers also face a lot of challenges that need to be tracked down. The aim of this webinar is to discuss those challenges and perspectives from multiple stakeholders. We have three speakers today: Dr. Yiming Zhang from Huawei Technology, Dr. Ravi Kara from Labcorp, and myself on behalf of Median Technologies. Labcorp is a leading global life science company that provides vital information to help doctors, hospitals, pharmaceutical companies, researchers, and patients make clear and confident decisions. Through empowered diagnostics and drug development capacities, Labcorp provides insights and accelerate new innovations to improve health and improve life. Median Technologies is a leading AI-based next-generation precision medicine company. We focus on helping conquer cancer and other diseases through routine imaging tests and imaging service for drug development. With our office based in US, Europe, and China, we operate globally to deliver best-in-class end-to-end imaging service for your oncological trials. A practical reminder that you may submit your questions during the presentation in the question panel, and we will answer them at a roundtable discussion. Now, let's welcome our first speaker, Dr. Yiming Zhang. Dr. Zhang is the Vice President and Chief Medical Officer of Huawei Technologies. He was trained in China as a nuclear medicine physician with PhD in radiopharmaceutical science and postdoc in Sunbat Hospital in London. He also has been a medical fellow in German Cancer Research Center before he moved to US and became Associate Professor of Radiology. Being an excellent physician, professor, and researcher in academic hospitals, Dr. Zhang joined biopharma and has been working in the industry in the past 2 decades, including Millennium, Abbott, AbbVie, using molecular imaging to assess drug development. Dr. Zhang also served as an honorable president of the Chinese American Society of Nuclear Medicine and Molecular Imaging. Dr. Zhang, we have the privilege to have you today to open the discussion and give us a talk on valuable of modern imaging technologies in oncological trials. Thanks.
Thank you, Yan, for your nice introduction. And next, please.
Okay.
For tumors, I will share some of my experience. Sometimes it's a painful experience, both as an investigator and also as a sponsor. And now I'm trying to develop some new imaging agent for molecular imaging. So I will share some mixed feeling about the imaging in clinical trial. Tumor-seeking imaging usually is used as your imaging CRO as a housekeeping technology. This tumor-seeking imaging most of the time is anatomic, like a CT, MRI, even some functional MRI to form the diagnosis. And also when you re-examine the patient, you can get the re-diagnosis after some kind of intervention and the treatment to the patient. These are anatomic imaging usually forms the initial foundation of the diagnosis of a patient with a tumor and forms the foundation for inclusion into anti-cancer trials. For re-diagnosis is sometimes just used for the efficacy evaluation and for the RECIST, most of the time we measure the tumor response with anatomic imaging. Next, please. Sometimes only anatomic imaging is not sufficient, so we added a functional imaging using FDG PET. Usually, FDG PET also being included as a molecular imaging, but because its spectrum is so wide and not reflect a specific molecular event, so I can regard that as a functional imaging. Sometimes the functional imaging are included in the patient selection and also included for evaluating the patient response. Next, please.
Okay.
The real molecular imaging, most of the time we use a PET as an imaging tool for patient selection for some targeted therapy. Because in the targeted therapy, most of the time we establish the patient's molecular pathological diagnosis by examining the primary tumors archived from the early surgery. But as we know that when the cancer recurrent or metastatic disease, especially with multiple metastatic disease, it's different disease already. So pre-mapping the target expression to establish the molecular pathological diagnosis with whole body resolution is very desirable. So this is the power of PET. The early example and established example is using the estrogen receptor PET imaging, and sometime recently we have the somatostatin receptor PET, and also the HER2 PET, PSMA PET, as a oncological paradigm to amyloid beta PET imaging for Alzheimer's disease treatment. This forms the fundamental of inclusion. We're seeing the positive patients, we include them. And only seeing the positive, the target positive lesion, sometimes not sufficient for re-diagnosis tools. So for evaluating the tumor response, we still need the RECIST to measure the tumor size and even some functional imaging with FDG PET. Next, please.
Okay.
PET imaging for the patient selection is the best example by the anti-estrogen receptor imaging with radio labeled and fluorine 18 labeled the analog of estrogen, as shown on the right in the upper corner of the structure. And the left lower panel showed the difference of FDG imaging, which reflect the tumor biological and metabolic activity, and estrogen imaging, we call the FES. This is a tracer's name, FES. The patient 1 and the patient 2, patient A and the patient B, one is positive and one is negative. The right panel is a very established example for the anti-estrogen treatment. The left panel is before treatment. The patient 1, we see that with FDG, we found that recurrent breast cancer, and FES imaging showed the positive estrogen receptor expression. And this patient received anti-estrogen treatment. After 6 weeks, tumor activity, biological activity is reduced, and the size also reduced. The same FDG diagnosis for case 2, showing that the recurrent of breast cancer, but estrogen negative, and this patient become the victim of this the same treatment. Interestingly, the FES being used in hospitals for almost 20 years as an in-house imaging agent, and after almost 2 decades, it is approved last year, and now every hospital who has the PET scanner can use that because it is commercially available by manufactured by the central radiopharmacy and delivered to the hospital. The next type of the targeted imaging is we call it radiotheranostics. This is a pair. The imaging agent is radio labeled, and the therapeutic agent is also radio labeled. This is called the radiotheranostic. This pair don't have to be the same carrier molecule, exactly the same type of carrier molecule, but they must target the same target. Another thing is it is desirable that the diagnostic pair, the diagnostic part, either PET imaging or SPECT imaging, is already commercially available. If not, imaging agent or technology is currently applied to agency for approval prior to the treatment counterpart. This is a very time wasting and also painful. We are advocating in regulatory agency to allow us to applying therapeutic pair, theranostic pair at the same time, which will save a lot of time and cost because we can use the same patients. Next, please. I will continue to explain the radiotheranostics. Only seeing the target positive lesion usually not sufficient for inclusion or exclusion. Some lesions with lower target expression will escape the targeted radiotherapy and can contribute to the disease progress. In this case, we need to use the FDG PET to evaluate the lesion's biological activity and compare that and match that to target expression imaging, which is a powerful tool to exclude mismatched subjects, someone possible with growing lesions by receiving the targeted therapy. And a good example, you can find that the PSMA PET imaging and FDG PET imaging matching for lutetium PSMA 617 phase 2 trial conducted in Australia. And this trial is very interesting, and each year the pioneer and also the leading PI, Dr. Michael Hoffman, will give an update during the ASCO. And each time, sometimes the conclusion is a paradox. I continue to read. Only seeing the reduction of target positive lesion uptake, tracer uptake in the follow-up PET only reflect the target expression change or just the uptake reduction, usually not sufficient as a rediagnostic tool for response. For response, RECIST plus FDG PET, even overall survival are still the measure of the outcome endpoint. Next, please. This gave the example of the trial conducted in Australia for PSMA 617. You can see that the inclusion and also exclusion, they use the PSMA PET imaging and FDG PET imaging matching for the patient selection. Next, please.
Okay.
There are more tracers and treatment pairs coming. One example is called the FAP, fibroblast activation protein. This is a stroma, is present on the stroma, and is a pan tumor imaging agent, and people are developing therapeutic version of this radiopharmaceutical targeting the same lesions. Next, please.
Okay.
And move next. And this is the first in patient with a new version of we call it the FAP. This is they use a dimer to increase the binding affinity and also modify the pharmacokinetics. Next, please. The next, this is my last slide. I will conclude that selecting imaging technology into a clinical trial is very challenging and painstaking. And what we want to select and what is available depend on the clinical development stage. For early development, we like the sponsor like to have multiple and some more specific imaging modality or imaging technologies for tighter patient selection and achieving proof of concept conclusion more confidently. The later stage development, we need something more robust, easy to standardize. And the last challenge is working on the imaging charter. This is almost the most important part for a successful application of imaging in clinical trials. That also as a hub to connect the PI, patient, CRO, and site, and also the sponsor. The recurrency with multiple metastatic lesions, sometimes it's numerous or countless lesions, is very difficult for radiology to identify. That coming the time for artificial intelligence to add human intelligence. So our next speaker will addressing this and give us excellent progress and application. Thank you very much.
Thanks a lot, Dr. Zhang, for your excellent talk. And also, as a kind reminder to our attendees, you can keep your questions and you can submit your questions in the question panel, and we will answer that during the discussion session. Now I will welcome our next speaker, Dr. Ravi Kara. Dr. Ravi Kara is currently Senior Medical Director Oncology at Labcorp Drug Development and is based in Germany. Ravi is a physician with over 20 years of experience, including patient care, clinical research, product launch, and product lifecycle management from clinic to industry. And so Ravi has extensive clinical development experience in solid tumor across all phases with multiple target agents and immunotherapy. So Ravi, now the floor is yours. Thank you.
Thanks, Ian, for the nice introduction. And good afternoon, everyone, and welcome to this session on the challenges in clinical development of imaging biomarkers. We basically have two sections to cover. The first one will be what is the current status of drug development, and the second one will be on the challenges with new imaging biomarker trials. Let's take a look at what is the current status of drug development. Well, if you look at the FDA approvals of oncology drugs across indications in the 10-year period from 2006 to 2016, there has been a systematic and a heartening increase in the number of approvals. Now, if we dig deeper, we discern that priority reviews have been increasing year on year. Now, we know that priority review is reserved for drugs which, if approved, would be a significant improvement in the safety or effectiveness of the treatment, diagnosis, or prevention of serious conditions when compared to standard applications. That basically means that in this decade, between 2008 to 2016, the drugs approved by FDA met one of the aforementioned criteria, which can be generally translated into more effective and less toxic treatment for patients. If we look closer, we find that the vast majority of drugs and the biggest progress has been in the area of immunotherapies, whether it be active or passive. This field has shown that a lot of activity actually can mean progress, and currently there are close to 5,000 products in various stages of development, including T-cell targets, targeting modulators, cell therapies, so on and so forth. I'm confident that this trend will continue to grow for the betterment of patient lives. I'm trying to move the slide. Yes. Despite the science getting ahead by leaps and bounds, the methodology of testing and validating the science has not really caught up with the progress, meaning the development of drugs is mostly linear and sequential, with the traditional preclinical followed by clinical development steps of phase 1 to phase 3, with increase in the size of the trials and the duration of treatment associated with a preset regulatory milestones leading to an approval. Now, this worked in the era of chemotherapy, where drug development was mostly by serendipity. This model is unsuitable for the immunotherapy era, and there are several reasons. Number one, because of the long development timelines, access to promising drugs takes time. And the second reason, the longer the duration, heavier the financial investment. And third, and the most important reason, along the development pathway, patients may be exposed to drugs that may not benefit them, and in the worst case scenario, may end up harming them. Now, if this linear and sequential model is ill-fit for the era of immunotherapy, what is going to work? What is going to definitely work is the seamless design, which has already been proven to work by how pembrolizumab was developed. And this development actually feeds from the science and then feeds back into the science, meaning that we have a continuous and integrated development where we learn from the early clinical trials and then implement them in the late phase. What do I mean by that? I mean, we need a proof of mechanism and proof of efficacy as soon as possible, and that means that we entail trials, design and develop trials that are flexible, continuous, and integrated, and can generate data faster, and based on which the trial generating the data can be modified, or new trials can be set up based on the data. Now, just taking a closer look at how pembrolizumab was developed, we have seen that this was basically the first drug to be approved across indications in the microsatellite instability high, regardless of the tissue of origin tumors. And this was this drug was basically tested in multiple, at least 15 different tumor types. Now, what we definitely lack even among this immunotherapy era is that what we do not have a single biomarker that can robustly tell us which patients are going to benefit and which patients will not. Now we know that the traditional source and use of biomarkers, we have been using them as a prognostic biomarker or a predictive biomarker, but there has also been an increasing literature on how biomarkers can be used as to identify resistance or even as a pharmacodynamic biomarker. Now, what we definitely need is a biomarker that can actually tell us what's happening in the tumor in real time, and as far as possible, non-invasive. So what we definitely need is to have imaging, for example, biomarker that can tell us, number one, atypical responses, pseudoprogression comes to mind, or is reliably able to distinguish responders from non-responders, but most critically, visualize tumor microenvironment and changes in tumor composition before, during, and after treatment. What do I mean by that? What do I mean is that the need of the hour is we need to visualize what's happening at the cellular level. That is the picture on the left, reproduced by imaging, which is a picture on the right. There has been immense progress in that direction with multiple imaging agents, including very specific targeted ones like PSMA in the prostate have been developed, and there are several others that are being developed, as we have seen from the examples from the previous speaker as well. However, the development is beset with several challenges. Now let's take a closer look at what those challenges are from different stakeholder perspective. Now let's start where the real action is happening, that is at the site. The two key challenges are how to motivate patients not only to enroll but to retain them till the end of the study. This can be quite subjective and differs between investigators even in a single site. The second one is logistical, how to ensure all protocol related activities are done fully and within timelines, especially when patients need to be in the scanners for a couple of hours, which is very unpleasant to say the least. Now, both the aforementioned challenges can be managed and overcome when you have the right multidisciplinary team at the site. The team must include, apart from the treating oncologist, an imaging specialist, and preferably a dedicated nurse or other paramedical staff. Why? Because generally the paramedical staff are better in speaking to the patients and explaining details in a lucid and a patient-friendly manner. In my experience, I can say that the presence of a multidisciplinary team has dramatically altered the dynamics at the site, leading not only to increased recruitment, but also to the retention of patients in comparison to sites that really lacked a multidisciplinary team. When looking at the patient's perspective, it is a series of questions that we must be ready to answer. The first and foremost being, obviously, there is no therapeutic benefit for the patient. And the second, is it worth the time and the effort? Now, I think both these questions have a similar, if not the same answer. That is, we know that there is quite a bit of literature which shows that patients in any type of clinical trial actually do better than patients in routine care. Now, this is due to the rigorous application of the protocol specified, for example, schedule of assessments, the investigator and the site staff spending more time with patients to meet the demands of the protocol data collection, and so on. The last question is, even if the patient has enrolled into a trial, will they continue till the end of the study, which is of paramount importance if we want to have a complete set of data that will enable us to fully evaluate the study and the imaging agent properly. Now, this again boils down to the efforts of the multidisciplinary team at the site and their engagement with the patient. Now, moving on, Now, let us take a closer look at the other stakeholders, the sponsor and the clinical research organizations. Now, the sponsors need to have a very clear vision on what their objectives and end goals are. I've seen instances where sponsors end up with a drug that is approved by the regulators, but that's not reimbursed by the payers or not widely accepted or used by the physician community. Now, that is a real nightmare scenario, which can be pretty much negated if the sponsor, number one, engages with the right key opinion leaders and key external experts as a part of either an advisory board that is willing to collaborate and spread the science and the data in a balanced way. Number two, convincing payer data and clear pathways in first launch countries. Example, Germany is always a good country for first launch because of the straightforward market access requirements and pricing, but also the data requirements. If your data can fly in Germany, it will be pretty much accepted in almost all other major markets. Number three is all of the aforementioned can only be possible if you have the right collaborators, which brings us to the other stakeholders, which is the clinical research organizations like Labcorp. From the clinical research organization's perspective, we must have an extensive network of sites and investigators. In fact, at Labcorp, for example, we have our so-called early phase oncology network, which has access to top oncology sites in North America and Western Europe. And the other one is the CROs must provide adequate and continuing support to site and investigators, whether it be the physicians or the medics working on the project managers, or the clinical research associate or others who liaise regularly with the sites and have to provide them with the required support and guidance in the conduct of the study. And it also makes sense to have a patient recruitment and retention strategy for all of these imaging biomarker trials because of the aforementioned the challenges that we have seen. In fact, for example, at Labcorp, we do definitely have a department that is taking care of patient recruitment and engagement, and whose main objective is to provide tailored solutions for each study on how to best recruit and retain patients. Now, with all of this, it's always essential that you have a collaborator that is vested and interested in the success of your drug. This is for the sponsors. Always select the collaborator who's interested in the success of your drug as yourselves, because this is exactly sometimes what I would call as the making or the breaking point. Now, we have seen challenges with developing imaging biomarkers and also how they enhance targeted and precise patient management. But imaging can take us into the next level in patient care if we use artificial intelligence and data at our disposal, says our next speaker. It's my pleasure to introduce Yan Liu, the Chief Medical Officer of Median Technologies. Yan Liu is an imaging expert with 20 years in clinical and academic research, with the past 10 years dedicated to the integration of imaging in multi-center oncology trials and validation of emerging biomarkers. Previously, Dr. Liu served as the head of translational research, radiotherapy, and imaging department at the prestigious European Organization for Research and Treatment of Cancer in Brussels, Belgium. She has published multiple book chapters and more than 35 peer-reviewed papers in high-impact journals, and was also a member of the RESIST working group. Yan, the floor is all yours.
Thanks, Ravi, for a very nice introduction. And also, it's my great pleasure to share with you some of the thoughts on how to use AI-powered imaging to optimize clinical trial design and eventually to increase trial successful rate. We know that medical imaging is widely used in the entire life cycle of drug development. For example, using radio-labeled monoantibodies against CD8 positive T cells to target imaging of CD8 positive T cells in patients with cancer to test the drug PK/PD. In the past 2 decades, K-trans of DCE MRI has also been implemented for anti-angiogenic drugs, dose escalation, thanks to its quantitative measurement on vascular inflammation. A safety biomarker, left ventricle ejection fraction, is also commonly used as cardiac function biomarkers in TK inhibitors. All those novel imaging technologies, they can be used for proof of mechanism. When a drug is entering into phase 2, RECIST-based criteria often taken to assess drug efficacy, measured as objective response rate. And then results will be also confirmed in large phase 3 studies. And after that, we have the market authorization, and imaging modalities are still used for routine evaluation to assess treatment risk and benefit. So we know that at Median, in addition to this above values as standard service provided by ICRO, we also believe digital biomarker powered by AI could also take a key role in the future clinical research. For example, using machine learning technologies, we can build models to predict patient molecular profiling to better understand mechanism of action. In addition to the left ventricle ejection fraction, body mass index may also be used as an additional safety biomarker for dose selection. Using AI model, we can also predict the responder at an early stage to help drug developers to better make go-no-go decisions at early stage. For confirmatory trial, AI tool can also to reduce the reader variability and provide insights on patient pseudo progression. After drug enter the market, many data will be generated from real-life practice. So through data mining, they can not only validate AI models, but also provide incredible sources of information for future drug discovery and drug repositioning. So today I'm very happy. I will take lung cancer as an example to demonstrate how AI-powered imaging approach will redefine a new patient journey in cancer treatment. We know that lung cancer today is the #1 cancer killer globally, probably because the majority of the lung cancer patients are diagnosed late and when cure is no longer possible. So today, low-dose CT is used by the United States Preventive Service Task Force screening guideline, and it's also reimbursed by CMS. With the deep learning method, it has the potential to also increase accuracy, consistency, and adoptions of the lung cancer screening in worldwide. So Median uses iBiopsy platform to develop an AI-based algorithm, enabling the identification and also characterization of those nodules with the low-dose CT. It decreased the number of false positive and also decreased the overdiagnosis. This end-to-end CAD-E and CAD-X solution demonstrated already very high accurate performance with error and a curve above 0.90, even in early stage 1 disease. Today, Median's AI is also moving quickly into clinical validation phase, and hope that thanks to those AI tools, we will be able to capture lung cancer patients at early stage when the disease is still curable. Although surgery offers the best chances for long-term survival in patients with early-stage lung cancer, however, the long-term survival after surgery remains less than 50%, mostly due to the high tumor recurrence rate. In October 2021, FDA approved atezolizumab as an adjuvant therapy for stage 2 to stage 3A non-small cell lung cancer with PD-L1 expression greater than 1%. Now, adjuvant treatment are increasing, but who should receive those additional treatment? CD8 PET imaging has shown promising results in early phase trials. And also different treatment strategies were proposed to assess whether patient will benefit from immunotherapy. And if yes, which therapy we should give, monotherapy, combination therapy. However, we also realized that CD8 PET is not feasible for all centers. and also it is associated with high cost. Therefore, a potential alternative solution to predict lymphocyte infiltration based on standard imaging using AI could be useful. So Medium is working on a solution to predict CD8 positive cell tumor infiltration using deep learned features based on CT imaging. The data set from TCGA with 48 patients in liver cancer. And we also show a lower mean average error than the traditional radiomics features, and also with a more accurate predictive value of error in the curve 0.93. In addition, compared to radiomics, our biopsy technology is lesion-free, lesion segmentation-free, which also avoid time-consuming tasks for lesion segmentation. Besides this, we also tested in non-small cell lung cancer with 104 patients and achieved error in the curve around 0.7 to predict the CD8 positive cell tumor infiltration. Following all these very promising primary results, we are working on a large data set for optimization. When we talk about safety biomarkers, sarcopenia is a well-known prognosis biomarker for negative outcome in cancer patients. More than 15,000 records were found in Google Scholar when we searched sarcopenia drug toxicity. It influenced treatment, discontinuation, dose reduction. Today, chemotherapy calculation is still based on body surface area. Although this method has been used for many years, it has its limitation, as it does not take into account either individual differences in body composition or pharmacokinetics. It has been demonstrated that body composition has a strong association with drug dose-limited toxicity, which also leads to drug discontinuation, postponed treatment, or dose reduction. Body composition can be measured quantitatively via imaging methods, such as dual energy X-ray, and also a cutoff is defined for both men and women. However, dual energy X-ray is usually an additional imaging method, which adds logistic arrangement and also cost in a trial. Body composition index can also be calculated using fully automated AI quantification tools. Bone mass, fat mass, and lean body mass can be extracted with or without manual correction by human to provide quantitative analysis on body composition index so as to provide a reliable and personalized guidance on chemotherapy dose. Cancer patients also usually have very active surveillance on disease recurrence. So during this period, AI-powered imaging could also help to follow treatment monitoring and allow early and accurate detection of relapse. Medium biopsy lymph screening product may also be extended to monitoring disease recurrence. During the blinded central review monitoring, as medical monitor, we often observe that our readers may miss new lymph nodules when they're small, subtle. So at early stage, it is difficult to differentiate newly appeared small nodules between truly new metastasis of lymph or this is due to inflammation. Therefore, an AI-powered tool can help us to reduce the reader variability, thanks to the function, and also ease the differentiation between true progression due to new lesion or pseudo progression due to immune inflammation. For locally advanced non-small cell lung cancer, neoadjuvant setting with immune checkpoint is now also in the lung cancer treatment landscape. In March 2022, FDA has approved nivolumab for adults with resectable non-small cell lung cancer. So for those who have tumors larger or equal to 4 centimeters or with positive lymph nodes, in a combination with platinum-based chemotherapy. I'd like to share with you this very interesting example. These two cases were published in New England Journal of Medicine in 2018. Patient 1 is a 62-year-old man with stage 2B squamous lung cancer. After administration of nivolumab, the primary tumor had significant shrinkage with associated tumor cavity. It was a partial response according to RECIST 1.1. The second patient, patient 5, is a 78-year-old female with stage 3A lung adenocarcinoma. Now after nivolumab, we can see the primary tumor size increase. So apparently, according to RECIST 1.1, it is a non-responder. Interestingly, pathological results showed 100% pathological regression of the primary tumor for patient 1, and also 90% tumor regression, so major pathological response for patient 5, so the patient B. So those two cases are responder cases. The assumption of the tumor size increase was probably due to infiltration of immune cells into tumor. So from this example, we can also see the limitation of conventional images to assess immuno-oncology treatment. Here is a very nice example. The publication from the group of Dr. Schwantz demonstrates that machine learning can identify a combination of quantitative radiomics imaging features, which predict overall survival better than RECIST 1.1 in patients with melanoma treated with immunotherapy. Those machine learning tools can potentially identify responders or non-responders to guide treatment at early stage. So it could be particularly helpful in neoadjuvant setting to stop preoperative treatment for non-responders and to avoid any delay of operation. Despite cancer program, the screening, and also dynamic changes in treatment, many patients are still stage 4 and need to receive symptomatic treatment. AI-powered imaging approach could also help to understand patient-specific profiling to guide adaptive deployment of combination therapy. As we know today, more than half of cancer patients do not benefit from immune checkpoints, and the combination treatments suffer from significant limitation, including serious toxicity. At the moment, there was no single biomarker has both high prognostic or predictive value, probably due to complexity of tumor heterogeneity. And here, non-invasive imaging biomarker powered by AI may play a very important role. Unlike tissue samples are taken from a small proportion of the tumor, non-invasive imaging biomarkers could provide insight on the entire organ, including tumor heterogeneity, tumor microenvironment, and host environment. So those imaging clusters could potentially identify responders and non-responders to anti-cancer treatment. So in this regard, AI-powered imaging methods could be valuable to optimize a clinical trial design and can also help drug developers to better understand the underlying histopathology, tumor microenvironment, and to predict who may benefit from immunotherapy. And thanks to these values, those tools could stratify patients to be treated with monotherapy or combination therapy. In summary, we believe that AI-powered imaging approach provide additional insight for trials. Through those new technologies, we could better select patients for trials to increase successful rate, to better predict response, and also eventually to help drug developers to make go-no-go decision, to accurate monitoring for disease relapse, and also provide a great solution for safety assessment. So thank you. This is what I would like to share with you. So now maybe I can open the discussion session. And I will also invite our two speakers to join. Dr. Zhang, could you please join as well? And now I have some questions that I would like to share with you. Okay, I got a first question that's for Dr. Kara and Dr. Zhang. So, Dr. Kara, today most diagnostic radiopharmaceuticals, except FDG, they are used in early phase. Do you think that they can be used in late-stage pivotal studies in the future? This is a question for both.
Okay, who take it first?
Dr. Zhang, you can.
I try. This is a nice question. I try to give some interpretation about the feeling and what I have found. For some radiotherapeutic and also for targeted therapy, for example, if you develop a new anti-hormone treatment for breast cancer, you can use FES PET. That definitely, and even using in the late phase trial for select more suitable patient to treat. But for some, it's difficult, and for using the new molecular imaging, the best scenario will be in select patient, but not for monitor and response. For monitor and response, still use the anatomic probably is more suitable. Even sometimes you can include the FDG PET.
Okay, interesting. So from your opinion that it can be used for patient inclusion, but using monitoring tool, it could have some difficulties regarding the feasibility and the cost considerations. So Ravi, do you have other opinions on that?
Not only the feasibility, because using that for example, for PSMA radiotherapeutic, if you see in the second round or later round of, because you're getting a multiple round of treatment, if you can using during the follow-up or monitoring imaging with PSMA, you see the lesions, some lesions getting dimmer, the uptake lower, doesn't necessarily reflect that tumor activity is low, the cells are killed. Maybe just reflect that the target expression is low.
Okay, great, noted. Okay, great. So Ravi, do you have anything to add?
No, I just want to, I first and foremost, I concur with what Professor Zhang said, and I would just like to add that we always need to factor in the logistics of getting this new tracers and imaging agents in a phase 3 trial across geographies and across multiple sites. Number one would be, is the site ready to perform, or the site personnel sufficiently qualified, trained, confident? That's number one. And number two, and the major hurdle still I see is the supply chain issues, and I have seen this practically in at least 1 or 2 trials that I have done with the novel agents. But I think it's only going to get better as more and more companies get involved into manufacturing of this new agents, as well as the better logistical distribution and supply chain issues are sorted out.
For the logistic supply, sometimes you can refer the patients to some imaging center and getting the result come back for you to evaluate the eligibility.
Mm-hmm. Mm-hmm. Yeah, and also indeed, once this imaging method is being covered by reimbursement by the healthcare, so there was no investment from industry side, so there was more suppliers, that will also promote the use of those novel imaging modalities. Indeed, this is really, there were multiple stakeholders will promote such kind of use to accelerate drug development and also benefit of patient treatment. I see there was another question coming. And it's for Dr. Kara, for Ravi. So how do you think that we can motivate patients for those imaging technologies? And especially when those imaging procedures are complicated, as you mentioned.
Well, that's a good question, and I think that's the crux of the issue and the main, I would say, rate-limiting step in any imaging trial that we have seen. At least I have seen in my personal experience. One of the first and foremost is to assemble the right multidisciplinary team at the site, as I alluded to during my talk in one of the slides. The key is the first point is to have the well scoped out multidisciplinary team that not only includes the physicians but also the paramedical staff, like a trained nurse, and so if we have a patient volunteer, so on and so forth, who have the time and the patients to sit with the patients, explain to them in detail what is going to happen to them, when, how, and how long. Once the patients really understand how they are contributing to the study and how the study is going to help them in a very minor way, because these are all not therapeutic trials, I think most of the patients will be happy to participate. And this talking with the patients and engaging with them should actually continue throughout the duration of the trials, to ensure that the patients will not only enter the trial, but they also are part of the trial till the end, so that we can have a solid data set that is robust enough to let us conclude on the outcomes of the study. So I think the first and foremost is having a very strong multidisciplinary team at the site. But also there are other things that could be done, like what Dr. Chang alluded to. If the site itself does not have facilities, the patient can go get the imaging done from another site. But here we could also have chaperone services that happens in several countries, where the patient is guided to the center and back, and the entire visit is coordinated and pre-planned, that the patient is not left to fend for himself or herself. So these kind of small logistical issues will also go a long way in making sure that the patient, once after entering the trial, is still motivated and committed to stay on the trial till the end of it.
Definitely. That makes a lot of sense. I see that we are closing to 1 hour, and I will suggest that I can move to the summarize of our 3 presentations. So we see that new imaging technologies are appearing to accelerate new drug development in the area of precision medicine. And thanks to Dr. Zhang and Dr. Kaveh's presentation, we can see molecular imaging is perceived as a tool to understand the PK/PD to support go-no-go decisions in early phase. On the other side, we also see that standard images also provide additional insights to show the tumor heterogeneities, microenvironment, thanks to the new AI technology, which allowed us to better understand the molecular pathway, the patient outcome. And as also during the discussion session, we understand that despite all those exciting points, we should not ignore the challenges linked to these new technologies. We should really to have physicians' adoption, be well prepared, considering technical feasibilities, and also, most importantly, to have patients' willingness and engagement. And the patient should be well informed about imaging procedures, their commitment, and any time, we should always put patients in the center. So thanks a lot for all your attendance and your time for today's webinar. And due to limitation time, we may not be able to answer all your questions, but no worries. We will answer your questions by email. And if you have further questions, you can also send that to info@Median Technologies. The replay will also be available at our website in the coming days. We welcome you also to join our next webinar on the topic of pinpoints with sites, imaging in the area of pandemic forced decentralized patient visits. So very looking forward to meeting you again. Thank you. Bye.